Okay, so maybe I can start with a little presentation of the group that is coordinating these webinars, and then I will let the floor to Margaret. So hello everyone. This webinar is the second one from a series that is coordinated from the SCOR working group for the BGC 168. That is about the coordination of the development of gridded four-dimensional data products from biogeochemical Argo observations. So we co-chair this group with John Sharp from US that unfortunately cannot be here today. And we have the pleasure to have Margaret Stoll that will give a talk today. So this group is composed of 21 members from 14 different countries. And the mission of our group is to facilitate the discussion and the coordination among the different scientific communities around the development, the validation, and the distribution of 4D BGC products from observational datasets, and with a specific focus on the BGC Argo array. So finally, the goal of this initiative is to significantly enhance the access and the utility of BGC Argo observations through the 4D BGC products, and thus finally refine our understanding of ocean biogeochemistry, improve models and reanalysis products, and inform policy decisions. So you can see the group that has met the first time during the OSM meeting in February 2024. And we hopefully will have our second meeting during EGU this year. It was just here to mention that we are gathering the products in a new web page that will be available through the BGC Argo website soon, officially. So in this web page, you will have access to all the BGC Argo database products. So it was here to showcase that it will be arriving. Here it's the link to the website of the product where you can find the group information, the group activities, the resources, and so on. And for example, the news that give, for example, information of the upcoming webinars. And also, please don't forget to sign up to receive the updates on the activities of the group on the Google form that you have here, and to have also the upcoming webinars that will arrive soon. And all these webinars will be recorded and then posted on the YouTube page, so they are visible after. So now I give the floor to Margaret, and thank you again to be there.
Awesome, thank you. Let me just pull up this presentation. Okay, can everybody see slides, no notes?
Yes, it's perfect.
Wonderful, thank you. Okay, hello everyone. Thank you for the introduction and thank you all for being here. My name is Margaret Stoll and I'm a graduate student at the University of Washington. And today I'll be talking about bridging ocean biogeochemistry and fisheries, so perhaps a bit of a different application than many of us are used to hearing about. I'll be talking about this connection between ocean biogeochemistry and fisheries under the umbrella of utilizing new tools such as these 4D BGC Argo-based data products to progress our understanding of ocean ecosystems. I'll also add a disclaimer that I am no expert in fisheries. I come from a background in biogeochemistry, but this interdisciplinary project provided the opportunity to work with a large team of wonderful people, some with expertise in fisheries, others with expertise in BGC Argo, and others in machine learning. So a big thank you to my co-authors for their support of this work. Oops, So to start with a bit of background, all of us here know that BGC Argo floats have really revolutionized our view of the subsurface ocean, both spatially and temporally, and enabled the study of biogeochemical processes and variability from a new perspective. So these BGC Argo floats are equipped to measure a variety of biogeochemical properties, including pH, oxygen, nitrate, biooptics. And the robust distribution of these observations has also facilitated the creation of four-dimensionally mapped data products. So these time-resolved, depth-resolved data products mark a notable shift in our ability to investigate fundamental biogeochemical processes concerning productivity, deoxygenation, and marine ecosystem health. There are countless ways to leverage these data products to study oceanography, many of which I'm excited to learn more about through this webinar. But today I'm going to share a somewhat different application that bridges to fisheries and fisheries management. So this research really focuses on the ways in which these 4D biogeochemistry data products can be leveraged to characterize. Sorry. uh, this research focuses on the ways in which these data products can be leveraged to characterize environmental conditions experienced by migratory marine species and in turn inform species distribution modeling. So species distribution models, or SDMs, you might see in the notes on the slides, these are models that predict the distribution of a species across space and time. And they can be utilized for a variety of purposes, from identifying suitable habitat for different marine organisms, all the way to informing large-scale ecosystem models. Now, the biogeochemical landscape in the ocean imposes critical constraints on suitable habitat. So marine organisms have varying temperature thresholds, low oxygen levels can lead to hypoxia. Similarly, acidification or high PCO2 levels can lead to hypercapnia, and biological activity serves as a control on feeding behavior and foraging conditions. So given that these biogeochemical characteristics kind of bound suitable habitat, their inclusion in species distribution models provides a basis for more reliable mechanistic relationships between species and their experienced environment. So I mentioned that species distribution models can be used for a variety of purposes, and they're quite instrumental in the world of fisheries and fisheries management, more specifically. So fisheries are biologically, culturally, and economically important. In the United States alone, fisheries bring about 1.7 million jobs and hundreds of billions of dollars in sales and value added to the economy. Worldwide, over 3 billion people rely on fish and other seafoods to provide substantial nutrition and protein. So understanding the role that biogeochemistry plays in dictating fishery species movements and how these relationships may shift in a changing world and climate is really important. So this work overall aims to assess the utility and value of these new BGC Argo-based depth-resolved data products to inform fisheries distributions. And to do this, we use juvenile North Pacific albacore tuna as kind of a case study organism, given their highly migratory nature and economic value as a US fishery. Albacore can be classified into 3 migratory subgroups, including long migratory, short migratory, and resident fish. The long migratory albacore undertake extensive seasonal migrations between the subtropical North Pacific during the winter and spring, and the coastal temperate ecosystems of the California current during the summer and fall. So this video below is just showing a single fish track and their seasonal migration. They cover quite expansive ranges over the course of a given year. The short migratory fish follow the migratory timing of the long migratory fish, but don't traverse quite as far west. And the resident fish decide to hang out off the coast of Southern California and Baja California year round. So not a bad life. But in addition to their quite expansive horizontal range, albacore also exhibit quite energetic diving behavior, frequently dive to depths beyond 200 meters during the day. And it's actually been estimated that cumulative vertical travel for juvenile albacore is between 8 and 12 kilometers, depending on the biogeographic regime. So their extensive horizontal and vertical movements cause this species to experience a really broad range of environmental regimes with distinct biogeochemical conditions. So to assess the utility of depth-resolved biogeochemical information in describing albacore habitat utilization, we use machine learning classification models that predict the probability of a fish presence or absence given some subset of environmental predictors. So the environmental predictors that we use for each model include a subset of temperature, oxygen, and proxies for optimal foraging conditions, including chlorophyll and particulate organic carbon. And I'll provide more detail on those specific data products that we use in the next slide or two. We use the XGBoost classifier as our machine learning algorithm. This is a decision tree based ensemble learning method. And our models produce a probability of finding a fish from these environmental predictors. And I also just want to note, for the purposes of this study, when I say biogeochemical information, I mean these three variables specifically. We know that temperature is a very strong predictor of fish movements, so we're really interested in determining where, when, and how biogeochemistry modulates habitat utilization. Okay, just to share a bit of background on our data sets. The albacore tuna data comes from two tagging programs that tag and release fish. These tags are attached to marine animals, and they have tiny computers inside of them that record and store data such as the ambient water temperature, body temperature of the fish, the location, and dive depth of the fish. And you may be wondering how these tags are retrieved. And it turns out that these programs actually post a bunch of wanted posters for these fish at marinas across the Western US coast, which I find pretty hilarious. These signs have information on the monetary award, how to remove the tags, who to contact, etc. So this gives us a very rich data set of presences across space and time. So here we're looking at a heat map of the number of presences in each grid cell, where the darker red colors indicate either multiple fish in that grid cell or a single fish spent multiple days in that location. Now, if a model is to predict a fish presence or an absence, you also have to teach the model where the fish did not go. And since it's not customary for fisher people to report where they went fishing but returned empty-handed, we have to generate this absence data set ourselves. So these are called pseudo-absences. There are a number of methods used in species distribution modeling to generate these pseudo-absences, which I'd be happy to talk about. But for the purposes of our study, the optimal strategy is random sampling from the background domain. So our machine learning model was trained on a balanced data set of presences and pseudo-absences. And here I'll just briefly introduce our data products, all of which are informed by Argo or BGC Argo float data. So the temperature data is derived from Romick and Gilson 2009. It has monthly resolution and is depth resolved to, I believe, 2,000 meters, which is far deeper than our fish care to venture. The oxygen data product that we use is called GoBio2, and it was created by our very own John Sharp and others at NOAA PMEL. This is a time resolved and depth resolved machine learning based data product that spans 2004 to 2023 at monthly resolution. And we also use two depth resolved data products of POC and chlorophyll from CMEMS, the Copernicus Marine Service. And these products use the SOCA method described in Sade et al. 2016. And this method merges satellite ocean color with Argo data to infer the vertical distribution of a variable of interest. And in this case, we're using POC and chlorophyll. Okay, last nitty-gritty methodology slide, and then we'll move on to some results. So we assessed the performance or the skill of our models using multiple evaluation metrics. We calculated the validation accuracy, the F1 score, and the area under the receiver operating characteristic curve. All of these are kind of a mouthful. The AUC ROC is a pretty common metric for binary classification. But for those of you not as machine learning or statistically inclined, values closer to 1 are better. And that's really all you need to remember. Okay, so those are kind of the main tools and methods that we use to explore the utility of these depth-resolved data products in species distribution modeling. So with that background, I want to introduce the main questions that this research aims to address. So first, we're interested in exploring the value of subsurface information in predicting albacore habitat use. And to do this, we created two-dimensional species distribution models. So something that looks like this, meaning the model predicts the probability of occurrence or of finding a fish in latitude, longitude, and time. And the two species distribution models are trained on different data products. So one is trained on surface satellite data, while the other is trained on depth integrated data informed by floats. And comparing the performance of these two models allows us to understand whether albacore movement patterns are in some way constrained by subsurface information. Next, if we find that subsurface information does indeed improve model performance, then we want to understand how different biogeochemical variables modulate habitat utilization. And to do this, we constructed a three-dimensional species distribution model that predicts probability of occurrence in latitude, longitude, time, and depth. And we analyze the influence of different environmental predictors on modeled suitable habitat across space and time. Okay, so finally, moving on to some results. So again, our first question involved an assessment of the value of subsurface information in predicting albacore habitat utilization. I mentioned that we compared two different species distribution models to address this question, one informed by surface satellite information and the other informed by depth integrated float-based data. And while surface conditions can sometimes serve as proxies for subsurface conditions, other times surface and subsurface conditions can decouple. Valuable information is not retrievable via satellites. So here's just one example of several fish tracks from the month of June, overlaid on maps of surface chlorophyll observed by satellites on the left, and depth integrated chlorophyll to 200 meters depth, informed by floats on the right. So we see that the integration with depth really changes the spatial pattern of chlorophyll hotspots. So we're interested in understanding how fish migration patterns may be driven by subsurface signals that aren't captured by satellites. So we compared the performance of these two different models, which are trained on similar environmental predictors. Satellite model was trained on SST, surface satellite chlorophyll, and surface satellite POC, whereas the float model was trained on SST and depth integrated chlorophyll and POC. Okay, so first, taking a look at the habitat suitability predictions from our float model, or the model informed by the depth integrated variables. So these plots are showing the modeled mean probability of a fish occurrence, separated by season. So values closer to 1, or the red colors indicate a higher likelihood of finding a fish. Values closer to 0, or the blue colors, indicate a lower likelihood of finding a fish. And the float model generally captures albacore habitat utilization and seasonal migration movements across space and time. This is also captured by our global evaluation metrics, which demonstrate that the model is fairly skilled, especially considering that we're only using 3 predictor variables here. So we were next interested in assessing the performance of the satellite-based model. So the model trained on surface chlorophyll and surface satellite POC data, as opposed to this depth integrated information. So here we're taking a look at modeled probabilities of occurrence again. The left plots here are showing predictions from the float model, whereas the right plots are showing predictions from the satellite model. And I only am showing two seasons here to not overwhelm folks. We have fall on the top row and spring on the bottom row. And in general, we see fairly similar patterns of habitat suitability between the two models. But divergences between the two models become more apparent when we take the difference between the two modeled probabilities of occurrence. So here we're looking at the difference between probabilities of occurrence between the float model and the satellite-based model. Orange colors indicate that the float model predicts a higher probability of occurrence in that region, whereas the purple colors indicate that the float model predicts a lower probability of occurrence in that region. So we see in the fall, the float model predicts lower probabilities of occurrence kind of off the coast of the California Current. But in the spring, the float model predicts much higher likelihoods of a fish presence in the kind of northeastern subtropical Pacific compared to the satellite model. So I'm going to zoom in on one of these regions and seasons, where we see some of the largest differences in predictions between the float and the satellite model. So again, in the spring, in the northeastern subtropical Pacific, we see the float model predicts a higher probability of occurrence indicated by those orange colors. So first off, we want to know if this increased likelihood of finding a fish, indicated by the float model, is actually accurate. So if we overlay the locations where only the float model accurately identified a fish, we see that they overlap quite nicely with the region where the float model actually predicted a higher likelihood of presences. So all of these locations shown here are presences that the satellite model failed to correctly identify. And We performed a regional analysis in this area and found that the float model was 25% better at identifying fish presences compared to the satellite model. So a fairly substantial improvement. So we were next interested in understanding which environmental predictor was responsible for driving these differences in modeled predictions. And to do this, we calculated the correlation coefficient through time between the surface satellite data product and the depth integrated float data product for each grid cell. And we did this for both chlorophyll and POC to understand where surface conditions may be decoupled from subsurface conditions and whether this pattern might be related to regions where the float model outperformed the satellite model. So here we're looking at the correlation coefficients between the surface satellite and depth integrated data products in the spring. So red colors indicate that the two data products are highly positively correlated, whereas dark blue colors indicate that the two products are anti-correlated. And again, the black circles denote locations where only the float model accurately identified a fish presence. And while we see relatively high correlations at these locations for particulate organic carbon, we see no correlation or weak anti-correlation between the two products for chlorophyll. So these regions could be telling us something about, for instance, interannual variability in the seasonal mixed layer depth, which could promote a sort of boom or bust in terms of productivity, depending on the year. So this kind of decoupling between the two data products could be driving the differences that we see in model predictions between the float-based data and the satellite-based data. So in addition to these environmental variables influencing modeled predictions of suitable habitat, we were also interested in exploring fish behavior in this region that could be driving one model to outperform the other. For instance, if the float model correctly identified fish that were actually diving deeper in the water column, that could indicate that the fish were targeting optimal foraging conditions that only subsurface information could capture. So we compared the diving depth of the fish that were accurately classified by the satellite model versus the float model. So here we're looking at a violin plot of the average daily diving depths for fish that were accurately classified by the float model in dark green and those that were accurately classified by the satellite model in light green. And we find that the float model accurately identified presences where the fish dove deeper in the water column. This was also a statistically significant difference. We expanded this analysis to all seasons and found that this statistically significant difference between dive depths for the two models holds. So these findings could indicate that the subsurface information incorporated into the float model does help to constrain and explain albacore habitat utilization. So just taking a step back for a moment to summarize our findings from this first research question. Again, this question aimed at assessing the value of subsurface information in predicting albacore habitat utilization. And to do this, we created two species distribution models, one trained on depth integrated information, the other trained on surface satellite information. And we find that the value of subsurface information on predicted habitat suitability does vary, but improves presence or absence classification accuracy in particular regions and seasons. We also find that the float model outperforms the satellite model where the fish dive deeper in the water column, which could indicate that subsurface information is valuable in describing albacore movements and behavior. So now that we've determined that subsurface information is indeed valuable in our species distribution models, we next created a three-dimensional species distribution model that predicts suitable habitat in time, latitude, longitude, and the depth dimension to understand how biogeochemical information kind of shapes albacore habitat in 3D space. So again, we created this 3D species distribution model, which was trained on sea surface temperature and now depth resolved temperature, chlorophyll, particulate organic carbon, and oxygen. So here we are looking at modeled probabilities of occurrence in the winter and summer. Again, red colors indicate a higher likelihood of finding a fish, greater than 50% chance of finding a fish. And we see some notable patterns emerge that align with behaviors we see in the fish data. So again, fish tend to migrate westward in the winter and reside in the subtropical gyre, whereas they tend to reside in the California current coastal regions during the summer months. We also see that the fish dive much deeper in the winter and shallower along the coast in the summer, which aligns with diving behaviors observed in the fish tagging data. This model also produced skillful evaluation metrics. Again, values closer to 1 are better. So this is a fairly skillful model, again, especially considering that we're only using 5 predictor variables here. So we can further understand this 3D habitat utilization by looking at sections of fish diving depth and modeled prediction of occurrences along its track. So here we're looking at mean daily dive depths of a single long migratory fish over the course of almost 2 years. And we see this pattern where the fish, again, dive deeper in the subtropical North Pacific, whereas the fish reside in the upper water column along the coast in the California current during the summer. And we can plot the modeled predictions of probability behind this fish track. And we see that the model does a pretty darn good job of predicting where the fish, on average, resides in the water column. Mind you, this fish track was left out of the training of this model, so the model has not seen this fish data before. So we can decompose these predictions into different combinations of predictor variables and ask, for instance, how does temperature alone constrain vertical habitat use? And we can take this one step further to understand where biogeochemical information specifically, so chlorophyll, POC, and oxygen, influence the probability of occurrence. To do this, we can take the difference in modeled probabilities of occurrence between these two plots to really extract the value of that biogeochemical information. So taking a look at the difference in probabilities of occurrence between the two model configurations that we saw on the previous slide, we can see where the inclusion of biogeochemical predictors either increases or decreases the probability of occurrence along the fish track. So brown colors show location in the water column where biogeochemistry decreases the likelihood of a fish presence, whereas blue colors show locations where biogeochemical information actually increases the likelihood of presence. And what's really interesting is this pattern of biogeochemistry expanding or contracting suitable habitat is consistent across all of our long migratory fish tracks. And this pattern is linked to distinct biogeographic regimes. So if we sync the time series above with the swimming trajectory of the same fish, we see that the brown and blue colors align with distinct regions and times of the year. So the brown colors tend to appear in the subtropical North Pacific. This indicates that although the fish is within habitable temperatures, it's not diving as deep as it could because of some biogeochemical aspect. On the other hand, the blue and green colors tend to appear in the coastal California current, and this indicates that although dives deeper in the water column fall outside of the fish preferred temperature range, there are likely optimal foraging conditions in these locations that are driving the fish to dive deeper in the water column. So we expanded this analysis to the whole domain, or the entire North Pacific, to determine how these biogeochemical variables modulate habitat utilization on a larger scale. So again, we ran our model with two different combinations of predictor variables, one with temperature alone, the other with both temperature and biogeochemical variables. And taking a look at these modeled probabilities of occurrence, this is the same color bar as before, except I've masked all of the values below 0.5 or regions where there's a low likelihood of finding a fish. So we see here that biogeochemical information actually contracts suitable habitat in the winter, especially considering the depth dimension. However, in the summer, we see that biogeochemistry actually expands suitable habitat, again, largely with respect to the depth dimension right along the coast. We then calculated the volume of suitable habitat predicted by these models and find that the inclusion of these biogeochemical variables contracts suitable habitat by over 60% in the winter and expands suitable habitat by 25% in the summer. So these biogeochemical variables are quite influential and important to include when attempting to capture the factors that modulate habitat utilization. Now, to this point, we've been probing the impact of our three biogeochemical variables, oxygen, chlorophyll, and POC, kind of collectively. But we can use other machine learning tools, such as feature importance or predictor importance, to understand where and when each biogeochemical variable kind of individually is particularly useful in predicting suitable habitat. So here we're looking at the relative importance of each predictor across the entire domain. So the sum of each of these bars totals to 1. The higher the bar, the more important the feature. And we see that sea surface temperature and depth resolved temperature are unsurprisingly our most important predictors. Again, temperature imposes a really important physiological constraint on fish movements. But our three biogeochemical variables account for over 40% of the predictive skill of the model. We also see that the importance of these variables changes quite a bit with season. So biogeochemical variables are actually the most predictive in the summer, again, when the fish largely reside in the California current. These biogeochemical variables account for over 50% of the feature importance, and chlorophyll and POC each account for about 20% of predictive skill. We also see a large increase in the importance of oxygen in the fall, again, when the fish largely reside in the California current. And oxygen is actually the second most important predictor, and it accounts for over 25% of feature importance during this season. So I don't have time to dig into all of the details concerning these analyses, but I did want to show just some brief results from a case study in the California current region in the fall, where we see this increase in the importance of oxygen as a feature that predicts suitable habitat. So to understand the influence of oxygen in the California current region during the fall, we again ran our model with two different configurations of predictor variables to extract the influence of oxygen on predictions of suitable habitat. So we took the difference between a model trained on temperature and all of our biogeochemical variables and a model trained on all variables except for oxygen. So here we're looking at, again, a 3D plot of the difference in mean probabilities of occurrence. I shifted the orientation of the plot a little bit to get a better view of the kind of coastal California current region at depth. In general, we see that the inclusion of oxygen actually contracts suitable habitat. Oxygen decreases the likelihood of a fish presence. And over the California current region in the fall, the inclusion of oxygen actually contracts suitable habitat by about 15% in this region, largely considering the depth dimension. So we next explored seasonal oxygen data in the California current to kind of mechanistically explain this decrease in suitable habitat attributed to oxygen. And we actually find that oxygen in the upper 150 meters of the water column reaches its lowest levels in the California current during October and November. So here we're looking at a 3D view of subsurface oxygen along the US West Coast in the fall. And the red colors indicate the threshold value of oxygen tolerance that's been cited for albacore tuna. So these low oxygen levels shoal in the fall and kind of begin to encroach on suitable habitat in this region. So this is just one quick example to show the ways that these tools can be used to disentangle drivers of species movements across space and time. There are countless other case studies that can be used to understand migratory species movements within their environmental context. So overall, this work has demonstrated that four-dimensionally mapped biogeochemical data products can provide a new perspective to understand migratory patterns associated with environmental conditions. And again, these species distribution models can be used for a variety of purposes to avoid protected species bycatch, or avoid fish at different life stages that have different tolerances and sensitivities to biogeochemical variables. But these data products are quite new and exciting tools in the world of fisheries. They do definitely depend on the persistence and growth of the global BGC Argo float array. But there are really endless questions that can be answered using this framework, kind of bridging ocean biogeochemistry and fisheries and applying BGC Argo to ecological questions through the use of these new tools. So that's all I have for you today. Thank you all for tuning in and listening. I will take any questions if there's time.
Thank you very much, Margaret, for this nice talk. So if people have questions in the audience. Jérôme?
Yes, hi, thanks for the nice presentation. I just probably missed the point. You don't have the locations of the tags, recover the tags. So how do you, I could not understand how you build this pseudo absence, or how you infer the location.
So we actually do have the locations of the fish data. Is that what you're asking?
Yeah, you said because of I understood that because of the fishermen do not want to explain where they have been fishing, or
the fishermen don't report where they went to go find a fish but did not find a fish. So we don't have an absences data set that we can train the model on to teach the model, these are the locations where we found a fish, And these are the locations where we did not find a fish. We don't have a data set where fish do not exist. So we had to create that data set ourselves. And that was through the technique of random sampling from the background. But there are a number of different techniques that people use in species distribution modeling to create that absences data set, because that typically doesn't exist. Does that answer your question?
Yeah, thanks.
Catherine? Yes.
Hi, Mary Margaret. This was really interesting talk. Thank you so much for sharing your work with us. I was wondering if you had, so do I understand it correctly that you're using the BGC variables to predict the fish populations at the same time? And if that's the case, have you tried looking forwards to see like if you could use the BGC data in January to predict like springtime fisheries or something like that?
Right. So that's a good question. So our fisheries data spans from 2004, I believe, to 2013. And most of the modeled the data is fairly sparse in time. There are some gaps in a few years. The data set is also a little bit imbalanced in terms of seasons. So we tend to have more data in the summer than the winter, let's say. So I don't think I have quite enough data to say, like to forecast, I think in the way that you're describing. What I have played around with a little bit is using our model or running our model on data in the future, or in the future relative to the fish data that we have, so beyond 2013, and seeing how suitable habitat changes from, let's say, 2013 to now. So I've looked a little bit at, marine heat wave years up and down the coast of the US to try and see if we can see notable differences in suitable habitat during specific events like that. But I'm a bit hesitant to trust that data too much, just because the training of the model hasn't really seen marine heat wave years. It's really only been trained. There were some marine heat waves between 2004 and 2013, but some of our more extreme events were beyond that. So a little bit hesitant to trust the results of that too much, but those are kind of some of the more forward-looking predictions that we've played around with a little bit.
Yes, I'm not sure to well pronounce your name, the Marcus Robinson. You can go ahead.
Yeah, thank you for the talk, Mary. Really did appreciate it. It was really nice to see the connection between ocean biogeochemistry and fisheries. My question is, do you think that this model can incorporate like otolith microchemistry? Because usually you can use this, which is air bones in fish. They're kind of like tree rings, and they assimilate the ambient chemistry environment. But do you think that you can use the various microchemistry proxies like strontium or barium as a more direct way to compare that with the ocean biogeochemistry from the float data to kind of get a more accurate representation of migration and also habitat?
That's a good question. I'm definitely not as familiar with that data, but I guess theoretically, if you have time resolved and like spatially gridded data products, then I think theoretically you can incorporate any variable that you want in these models. So I guess my next question would be, does that exist for the variables that you're talking about? yeah, just all of the data products have to be spatially gridded and depth resolved to kind of interpolate that data to the locations of the fish presence or absence data. But that would be really interesting. I'll have to think about that a little bit more.
Yeah, most definitely. Really appreciate it. Thank you.
I have a question. So I was wondering the integrated depth that you use to derive your depth integrated BGC variables.
Mm-hmm.
So
200 meters.
200 meters, okay.
Yep.
And yeah, my question was, have you thought to try to integrate all the shape of the biogeochemical profiles? So I mean, instead of using this depth integrated mean, for example, for chlorophyll, using all the profiles because I was thinking of the areas where you can have a deep chlorophyll maximum that could really more constrain the presence over the layer.
Yeah, I think that's a really, really great point, and you're totally right. I feel like taking a depth integrated value does a disservice in some ways to the variability that we see over such a large domain, such different looking profiles, whether you're in the California current or the subtropics or et cetera. I haven't played around with kind of other ways to capture that data, but I really like your thought of like somehow trying to represent the profile shape in a metric and feeding that to the model. And I'll have to think about that a little bit more. But I like that thought a lot. And I do think, I mean, in some areas that does make such a difference in terms of fish behavior. So I would imagine that could lead to even bigger differences between the two models.
Maybe we can talk later because, for example, in the models that we use to derive these products of chlorophyll, we integrate actually the vertical distribution of temperature salinity by reduce the dimension. So maybe we can talk about it, but I think it could be
Absolutely. Yeah, I would love to.
interesting.
Thank you. Yeah, that would be awesome.
Yes, Marin?
Hi, yes. Can you hear me?
Yes, I can hear you.
Yes, Mike.
Right. Sorry. Yeah. Yeah. A fantastic talk. I was so excited to see the way you've been able to bring together the 4D data and visualize it so effectively. All techniques very dear to my heart. And I would love to. Yeah. I wish I was not what 7, 8, 10,000 miles away, and we could chat about the modeling process. One question about it that I have is how it deals with sort of temporal lags in things. I mean, and we're all aware that these guys aren't eating biogeochemistry; they're eating stuff up the food chain. And I don't know how this machine learning stuff will deals with the fact of you have to move in time from where sunlight and nutrient-rich water come together, and the coherence of that bunch of water, and it's the oceanographic drivers of how where it moves to, how these models can deal with that complexity. Because I don't understand the models. Maybe you have a feeling about how they might be reacting to that.
Yeah, that's a really good point. I don't have a great answer for you, is my short answer. I guess the models, well, I guess I have an unsatisfying but a partial answer for you.
Right.
I've seen and it sounds like you might be aware, a lot of species distribution models do represent time in some way as a predictor variable that I think addresses some of the aspects that you're talking about of this kind of temporal lag between, all of these factors coming together and either like coalescing to create suitable habitat or not. So there are different ways that models have incorporated time as a predictor variable to account for some of that, I think. We decided to not include time as a predictor, just given the seasonal migratory patterns of the fish. Time is also a pretty strong predictor of where the fish is going to be and when. And we were really interested in targeting the biogeochemical variables with these 4D products, so we did not include time as a variable. But I think, if our goal of this research was really to create like the most accurate and representative model for albacore tuna, which I think our goals were slightly different with this project, I think that incorporating that sort of variable could be really important for all of the reasons that you mentioned. So slightly unsatisfying answer, but hopefully that touches upon some of the things that you're talking about.
Sorry, I have a phone ringing in the background, so I don't want to have my thing on, but thanks very much.
Yeah, of course.
Marin, did you have a question?
Yes, thank you, Margaret. That was really cool to see the progress. Nice. Yeah, I have a question regarding the environmental factor and also like the product you use. Have you thought about using DIC, depth reserve product as well? I was wondering if your species could be like sensitive to hypercapnia or like pH related effects, and if you think that could be maybe useful addition or not.
That's a good point. That's definitely something I had in the back of my mind at the very beginning of this project. I haven't actually tried to incorporate carbonate variables into the model. My intuition is that those variables would not so much impact the fish directly, but more indirectly through food sources. But short answer is I haven't explored that yet, but it is a thought that has crossed my mind. So a potential next step.
I have another comment, if I may. In Copernicus, so in the same product, in November from this year, you will have available the light variables operationally. And so maybe it will be too late, but in 2026, you should have also available the PFTs in terms of micro, nano, and pico phytoplankton in this product. So maybe it could be a perspective, to try to see if phytoplankton community composition can drive or not the absence or presence of tuna. So
that would be awesome. Yes, I would love to do that. And you said 2026?
Yeah, for the PFTs, yes.
Okay. I will sit tight. Yeah, I think that would be awesome to incorporate into models such as this.
Are there any other questions? So thank you again, Margaret, for this really nice talk.
Thank you. Thanks everybody for tuning in. I appreciate it.
Yeah, thank you everybody, and we keep in touch through the mailing list for other next webinars. Thank you.
Awesome. Thank you.